We will discuss how we stain, how we color the bacteria, and what are different methods, what are different techniques and dyes or coloring agents which we use to identify or to stain. First, to stain the bacteria, and these staining will be helpful for the identification of bacteria, type of bacteria, and clinically, not only clinically, you can say in microbiology, these staining techniques are the backbone of microbiology. Why? The bacteria, the bacterias, they are actually transparent. Bacterial cells, they are transparent because their refractive index is similar as water. So pure water is colorless, so we cannot if water is there, so we can easily see through the water, because if the water, pure water, is colorless. So same bacterial cell, so bacterial cell is also colorless because it has the same refractive index. So that is why when we, without staining, if we want to check, if we want to see the bacteria even under a microscope, it is impossible to see the bacteria because bacteria will be invisible even though we are looking under a microscope, but still we not be able to see the bacteria. So for this purpose, to identify the bacteria, to look the structure of the bacteria, type of bacteria, we need some staining technique, we need some coloring technique, so that the bacteria can be visible or visualized, should be visualized under the microscope. So with the staining method, the bacteria can be seen and easily identified even under a light microscope. So this is actually the main purpose of these stainings. So most of the bacteria, they have they have the similar structures, except some bacteria we discuss about mycoplasma bacteria, acid fast bacilli. So for different bacteria, we use different staining technique because of their structure. So let's start. So bacteria, I have explained you, it has a same refractive index as water. So we cannot see the bacteria under the microscope without any staining method. So staining, the staining is the technique uh, through which we can color our bacterial cell. And to, before coloring the bacteria, we must have to fix the bacteria on a slide. So for microscopic evaluation of bacteria, when we look for a bacteria, when we want to make a slide of a bacteria, so bacteria first should be fixed and then it should be colored. And after coloring, after staining, the identification points or identification of the bacteria we can easily made the identification. So we can see the morphological structures, we can see the cell wall, we can see even with the microscope, we can see some small the large cell organelles in the bacterial cell. And through this staining technique, we can also preserve the bacteria for future use. For example, in your microbiology lab, I don't know whether you will have the lab classes or not, but in your microbiology lab, you will see, you will have the fixed slides. So those slides are what? Those slides are preserved slides. So if you make a good slide, you can preserve the slide for maybe years, for up to years. Okay. So staining, it depends, the stain of or the dyeing or a stain, it depends on the bacterial cell wall or the bacterial outer membrane. So bacteria is slightly negative charge at a neutral pH. So as a rule of thumb, or as a general rule, the bacterial, because they are negatively charged, so the bacteria the sorry we have two type of dyes, basic and acidic dye. So basic dye, it stain the bacteria, while the acidic dye or the counter dye, it actually stain the background. So let's suppose if you have a slide, let's suppose here we have bacteria. So to visualize the bacteria, although you have done the staining, you have colored the bacteria, but still you need some background color. You should need some background color, because the background color or the background staining, it will enhance the color of a bacteria, as well as it will enhance the structure of a bacteria. So that is why by using this acidic dyes, this is also known as the counter dye, it helps to visualize more better. It helps to visualize better. So that is why the basic dye is a main primary dye which help to stain the bacteria, because bacteria retain the basic dye, and the acidic dye, they are responsible for the background. That is why we can easily visualize the bacteria and its shape, and we can identify whether it is cocci, bacillus, spirochete, or what type of bacteria it is. Simple staining technique, although we have different staining technique, the first type of staining is a simple stain. In a simple stain, we we cannot actually differentiate different type of bacteria. Simple staining is a technique in which we add some staining or a dye color or a stain color just to color the bacterial cell. So simple staining technique or a simple staining is a technique in which we use only a single dye or a single stain to color the bacterial cell. So this simple staining will actually lead to coloring of all the cells. So it will color all the cells which are present on the slide. But the problem is in the slide on the slide we have a group of bacteria are here. So this stain will color the simple staining technique or a simple stain, it actually color the whole slide. So all bacteria will be colored as a same color. So all bacteria will have the same color. So all the bacteria will be same stained. So the drawback of simple staining is, although it's a simple method just to check whether this solution or this sample have bacteria or not, but the drawback of this method, simple staining, is we can't differentiate between different bacteria. So simple staining technique is a technique in which we just add some stain, some color on a slide, and then we look under a microscope. So this technique is the simplest technique, but this technique cannot differentiate different type of organism. But we have other staining technique. I'm talking about the simple staining technique or a simple staining. Okay, simple staining. I'm talking about simple staining. So simple staining is a technique to just to color all the bacteria. And what is a
From staining.
and what is the stain? Stain is actually the coloring agent. It's the compound which give color to the cell. So because of the coloring, because of this color, we can identify, we can easily see. And we have different kind of stain, different kind of colors, different kind of coloring agent. So different stain or different staining techniques can help us to identify the bacteria. But simple staining technique cannot differentiate different kind of bacteria. So staining technique used for the microscopy just to enhance the microscopic image, how we perform this staining technique. And I told you that in microbiology, this staining technique is a backbone of microbiology, especially bacteriology, because all the bacteria must be stained before looking under the microscope. So without staining, we cannot see the bacteria. So that is why staining, it is necessary part in bacteriology. So how we do a simple staining? Simple staining, I told you, like we just need a simple dye. So a simple staining technique is a technique to identify the presence of bacteria, but it use only one color, one dye. So all the bacteria will be colored as same. So the first process or the first step of simple staining technique is preparing a smear sample or preparing a smear. What is smear? Smear mean we place a bacterial solution or a patient sample on the glass slide. That is, we call it smear. So smear is actually a thin film. It's a thin film of a bacterial solution which is present or which we make on a glass slide. This is a glass slide which we use for microscopy. So the smearing first technique or the first step of all the staining techniques, the first step is preparing a smear. So how we make a smear? We can use a simple this like we have these airbirds. Actually, it's a cotton bird. So we can use a cotton bird, and we can dip this cotton bird in a patient sample. For example, patient sample may be a pus sample, maybe a blood sample, maybe any sample from the skin. Okay, so we take a sample and then we just swap or we can say, yeah, a swap on the slide. Which one? This cotton bird. So this is we call the smear. So first step is the preparation of smear. So how we make a smear? We need to take a glass slide, and then with the help of a cotton bird, we take a sample and just make a thin layer or a thin film of the sample on a glass slide. This is a first method, a first step of almost all type of staining. The second method, the second step that is also common for all staining technique, is the fixation. Once you prepared a smear, so smear, is it liquid? It's liquid form. So there is a chances that the bacterial cell, or they can float or they can move on a slide. So uh, it may give us some false result. Or another problem is when we add a dye, a stain on a glass slide, we know that or when we rinse this slide with a dye, so bacterial cell may be wash out may be washed out with the stain. For example, if bacterial cells are present here, so for coloring, now we have make a smear. We have already made a smear. Let's suppose we have made a smear. So we need to add drop by drop. We need to add dye. So when I add a dye is a liquid. It's a liquid. So when I put dye on this glass slide, so what will happen? Because it's a liquid, so if the bacterial cells are not attached or fixed with the glass slide, these bacterial cell will be washed out with the dye. So to prevent this washing out, the bacterial cells must have to be fixed. What do you mean by fixed? The bacterial cells should have to be attached or glued with the glass. This technique is known as fixation. So how we do fixation? For a fixation, it's very simple. We, for example, we have, uh, let's suppose this is a flame. Okay. For example, this is a flame. So we just pass this glass light on a flame, maybe 1 or 2 times. So the heat is can cause or the heat can cause the fixation of the bacteria with the glass slide. So when we pass 2 or 3 times this glass slide on the flame, so due to the heat, the bacterial cell will attach with the glass slide, so that after this fixation, when I add a dye, so this bacteria will not be washed out. The bacteria will not wash out. So this is actually a significance for fixation. So first technique, or if these are actually the simple techniques, that is a simple technique, simple staining technique. And simple staining technique, the first 2 steps are similar to all other type of staining methods. So first is smear preparation, a second is a fixation. So remember, you don't need to heat for a long period of time, or do not expose to the heat directly. Like flame is here, if flame is here, so you need to pass the slide like above the flame, maybe about 1 or 2 inches above the flame, not on the flame. Because if you pass on the flame slowly, and it should be fast, like this, should be fast, not slow. If you are slow, then the heat will kill the bacteria and there will be no result. So that is why this is actually the precautions you have to made for fixation. So heat fixation, this is known as heat fixation. So this fixation is where you can say common for all type of technique, because it is cheap, it is easy. And third, we have a simple staining, like we just need to add some stain, some coloring agent. So this coloring agent we can pour on a slide. This is a slide. So we can pour a coloring agent directly on a slide. So I told you that when we pour the dye on the slide, if the bacterial cells are not fixed, then there is a chances of washing out of bacterial cell. So that is why we need to fix the bacteria with the slide. So simple technique, we can use a different stains. We have this methylene blue, basic fuchsin. So these 2 dyes, these are the dye coloring agent. They are available in the labs, you can see. uh, so these coloring agents, they are used for just to provide the color contrast, especially we use for all type of organism. Then we also have some Loeffler's ethylene blue solution. It is also a stain, it is also a coloring agent, it is also a dye. But this Loeffler's ethylene blue solution we only use for certain bacteria like Mycobacterium tuberculosis. So just wait, we will discuss later about this solution. So this solution is actually a saturated solution of methylene blue in alcohol with 30 ml of KOH, and this is actually the how we prepare these solutions. So you don't need to remember, but just remember the names of the dye. Okay, Loeffler's ethylene blue, methylene blue, basic fuchsin. These are the simple stain. These are they are the simple stains. Then we also have a diluted carbol fuchsin. This is another dye for simple staining, just to color all the bacteria. And this carbol fuchsin, diluted carbol fuchsin, is prepared by diluting the Zn staining. We will discuss later about what is this Zale-Nelson staining. So it's a diluted strain, so which also used for coloring bacteria, and most commonly this type of diluted carbol fuchsin is used to for Vibrio cholera. So if you are suspecting your patient have Vibrio cholera, then we can, or to identify the Vibrio cholera in a sample, then we can use this strain. Another type we have this polychrome methylene blue. This dye, it helped to identify the Bacillus anthracis bacteria. Class, any question?
No, sir.
Okay. So simple technique, it's a simple stainings, very simple technique. Just first to make a smear. I told you what is smear. Smear is just we pour the solution or a sample on a glass slide and make a thin film. That is we call a smear. Once the smear is formed, let the smear to dry, air dry, and then fix the bacteria, fix the smear by passing on a flame. And after that, we add a dye and then look under the microscope. So this is a simple staining technique. Simple staining. So what is the main problem of this simple staining? Although it is very easy to perform this. So we can use basic and acidic dye together, very easy. But the problem is, because we are using only a single dye, then all the bacteria will be colored as same. So that is why it will be difficult for us to differentiate the bacteria by simple staining technique. For this purpose, we use differential stains. Differential stains are actually, what is differential stains? When we stain the bacteria, instead of using one strain, if we use one or two or more than two strain, then it will help us to identify or differentiate different kind of bacteria within a sample. So differential stain, like using one or two or more type of stains at a time, that will help us to identify the bacteria, as well as uh, it will also help to observe the cell morphology, shape of the cell. So this is also very important because differential staining is nowadays we utilize this differential staining technique. The most important and widely used differential staining technique is the gram staining. So gram staining, this technique, its names come after this scientist name, Hans Christian Gram. So he was the first microbiologist who actually identify the bacteria, who actually differentiate and classify the bacteria according to their staining, according to the stain. And nowadays in clinic, in medicine, in medical, we follow this gram staining. And according to this gram staining, it classify the bacteria into two type of bacteria. Due to the classification, due to this staining method, we can identify, or we can uh, actually divide the bacteria into two groups: gram positive and gram negative. So gram positive mean that those bacteria which retain the gram stain, that are known as gram positive. So gram positive bacteria, they look like after the gram staining, gram positive bacteria, they retain purple to blue color, or bluish to purple color, bluish to purple color. While the gram negative bacteria are those bacteria which cannot retain the gram stain. So these bacteria, they colored as pink or red to pink. So after this classification, now it is very easy for us to classify the bacteria, to identify the bacteria, and to classify and to classify the bacteria, identify the bacteria, and to uh, prescribe the drugs according to those bacteria. For example, generally, like as a general rule, almost all of the upper respiratory tract infections, for example tonsillitis, pharyngitis, ear infection, nose infections, these infections, upper respiratory tract infections, are caused by gram positive bacteria. So we use the drugs which kill this gram positive bacteria, while the bacteria which cause diseases in lower respiratory tract, like in lungs, GIT, urinary tract infections, they are mainly caused by gram negative bacteria. So we have drugs which only kills the gram negative bacteria. So look, just with the help of this gram staining, we not only able to identify the bacteria, but with the help of this gram staining, we can also able to prepare the drugs to kill these bacteria. Class, understood?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
So what is the basic staining principle? The staining principle, the gram staining principle, it depends on the bacterial cell wall. So cell wall of bacteria is made up of, cell wall of a bacteria is made up of
Peptidoglycan.
peptidoglycan unit. So what is the difference between gram positive and gram negative bacteria? bacterial cell wall.
Sir, it's thicker. It's thicker and one is thinner in gram-negative bacteria.
Yes, very good. Cell wall of a gram-positive bacteria is multilayer and it is rich with peptidoglycan unit. Rich with peptidoglycan unit. While the gram-negative cell wall, it is thin, single layered, and it also have a outer membrane. While the cell wall of a gram-positive bacteria, it is thick and it is the only a membrane which is present outside the plasma membrane. So that is why, because the cell wall of a gram-positive bacteria is thick, so once the coloring agent enter into the peptidoglycan, it remain attached with the peptidoglycan. So when we add a primary dye, so primary dye will attach with this peptidoglycan. Even if we wash with some decoloring agent, but this primary dye will remain within the cell wall. That is why the primary dye color will remain at the end of this procedure. And because of this, the gram-positive bacteria will have a purple or bluish purple color. While gram-negative bacteria, their cell wall is very single layer. It's small, it's thin. So when we add a primary dye, so primary dye attach with the peptidoglycan unit. So first step, it will colored. The cell wall become colored. But when I add some decoloring agent like alcohol, acetone, so it will wash the dye. When it will wash the dye, so now the bacterial cell wall, there will be no dye because dye will be washed out by the decolorizers. And next step is when we add a secondary dye, or it is also known as counter dye. So when I add a counter dye, so when I add a counter dye on gram positive, so it will not enter or it will not attach with peptidoglycan. Why? Because the primary dye is already attached. It is already occupied in the gram positive cell wall. So secondary dye or a counter dye, it will not be retained by the gram positive bacteria. While the gram negative bacteria, their dye has been washed out. So secondary dye, when I add a secondary dye, it will attain or it will attach with the secondary dye. And after that, the color of this bacteria, the color of this bacteria will be similar to the counter dye. So that is why gram positive bacteria are those bacteria which retain the gram stain, while the gram negative bacteria are those bacteria which do not retain the primary dye or a gram dye. So that is why they are known as gram negative. Okay class, we have less than a minute. After a break we'll continue. Any question?
Not yet, sir.
Okay.